So I'm going to present work that we have been doing over the past few years with Yasser, Ryan, also other people were involved. So the general, so the talk is divided in two parts. The first part will be a little bit general, and the second part will be more on, say, applying the ideas of the first part to neuronal activity and to extract information, relevant information from data. Okay, so the general issue is that if you are interested in understanding living systems, so somehow living systems, they interact with an environment and they have to form an internal representation of the environment they live in, okay? And so this is a general problem that applies. So you can see gene expression in a cell, you can see the immune system, you can see pretty much everything, the internal organization of living system as a representation of the environment in which they live. And the issue is to understand what is this representation coding for, okay? Typically, also in this conference, we see that in many cases, you address this issue from the point of view of input-output relation. So you have a subject, you observe the behavior, you record something from the inside, and then you try to figure out what is the functional relation between input and output. So one very successful idea of this is information bottleneck, but say tuning curves, and this is essentially the way in which we go about thinking about these problems. Now the problem is that sometimes we don't really know what is the input that corresponds to a certain output, okay? And we don't really know what that particular state of the living system, be it expression profile or neural activity or whatever, is really coding for. And so this is why we decided to step back and to think about this problem from the point of view of what we call featureless inference. Okay, so and we were not the first one to think about these systems in this way. So these are a few quotes from people in the past. So this particular Lucretius warned us about really thinking in terms of input-output relations, in the sense that the living system, as we see, they are not really engineered to behave in the way we see. This one is a quote very dear to Yasser, which is essentially saying that you should really listen at the data as they speak, and yet this one and this one, which is even there is a debate on who pointed these things out first, but I think probably this is earlier than this. And it says that when you really want to understand what is going on in a living system, then you should have a non-parametric perspective. Okay. Okay, so
the ears were fashioned long before a sound was heard. I think I take exception with one of these claims.
So my point of view is, so our point of view is that essentially in all these representations, essentially relevance is related to frequency. So, for example, if you think about the problem of recognizing faces, building a repertoire of faces for a baby, then of course this lady that she sees over and over again becomes more relevant than other people that he just sees very rarely. And to the point that essentially maybe he will start to realize that when this face is like this, it means something different than when she is like this. So in the end, this somewhere it will start to generate a free energy landscapes where all these points correspond to some free energy minimum. And essentially that what I want to discuss is essentially what are the properties of data that convey information about relevance, and what is the statistical mechanics of the representation that is formed inside this this learning machine. Okay, so the first thing, if we take a data set, and here I'm just taking a data set where these observations are configurations which are discrete configurations. There are n of them. And I'm considering a situation where these are just IID. Okay, they are just drawn independently from a certain, you can think of them as being drawn independently from a distribution. But indeed, say maybe there are observations which are done on a system, looking inside a system in the same, under the same conditions. Okay, and so you can think that they come from a generative model where there is this probability distribution that probably tells you something about this system. Okay, so now if you want to understand how much information, so the key question is that we want to address is how much information does this data set contains on the generative model. And so the first thing that you do is to compute an entropy. So if this is the number of times that you see a certain configuration S, this is the frequency, then this is an estimate of the number of bits that you need to describe a single point in the data set. Okay? Actually, it's the number of bits that you need to store your data set, one point of your data set in your computer. Okay? However, not all these bits are really relevant. Some of them are noise, and some of them instead tell you something about the generative model. Okay? And if you think a little bit about this, what you find out is that this number of bits can be split into two parts. One part which is the entropy of the frequency of the random variable, which is the number of times you see a random configuration. And the other one is essentially the entropy of the configuration conditional on the frequency. Okay? And this is noise, and this is instead useful information. There are many ways in which you could get to this conclusion, but essentially the simplest way of understanding this is just that the frequency is your best estimate of the probability of the generative model, so that is where information on the generative model lies. And so you could see that, say, the way in which your data is generated is by, grouping all configuration with the same frequency. And so The entropy of this distribution is what tells you how many bits of useful information you have. And so we give different names to these two objects. One, we call it a resolution. This is a resolution because it depends on how you define the states. You can define the state with a more or less with more or less detail, and this number will change. But once you fix this number is fixed by the data, is fixed by the system, okay? And so we call this relevance. Okay, so if you really believe in this, then one aside of this is that you can understand how many parameters you can estimate from a given data set. You know, if you ask yourself, if you have a parametric model and you compute the mutual information between your data set and the parameters, essentially you get the big term, number of parameters divided by 2 times log n, plus constants, okay? So if you say that this cannot be larger than the number of bits that your data contains on the generative model, you get an upper bound to the number of parameters that you can extract from a model. But this is just an aside. Okay, so as I told you, you can, in many cases, you can change the resolution, so the way in which you define your states. And so this is an example where we just run clustering on 4,000 stocks in New York Stock Exchange, time series, okay, with 3 different methods. I don't need to go into the details. But essentially, for every, once you fix the number of clusters, then you have a size of the cluster, which gives you the frequency of a certain label. And you can build this number and this number. And as you change the number of clusters, this point will move from this point where essentially every configuration is seen just once. You have n clusters, to this point where you have just 1 cluster, okay? And so depending on the algorithm that you use to do data clustering, you get different curves. And what we say is that essentially the method, or the idea is that the method that uses, that has a highest curve is the one that extracts more information on the generative process, okay? But in any case, you can see that here you have a general trade-off between resolution, how much you compress your data, and relevance, okay? So when you decrease by 1 bit the resolution, your information that you get on the generative model increases at a rate which is the slope of this curve, okay? And now you can ask the question of, yeah?
Could I ask, what's the, is there a likelihood model here? So it's not just a function of the counts, is it?
It's just a function of the counts.
So you could
so
just divide these
you take
400,000 into groups of 200.
Yeah.
And that would achieve your...
Yeah, so you have the data. It's just a function of how you define the state, okay? You just count how many times in your data set you see state S. And out of this, you build the degeneracy, how many times, how many states you see k times. And out of this, you build these 2 quantities, okay? It's just, there is no, so this is why it's featureless, okay? There is no assumption on features, there is no assumption on models, it's just the data that is speaking, okay? Okay, so now you can ask the question, okay, so let's assume that we are at a certain resolution with a data set of n points. How does the sample that gives you maximal information on the generative model look like? And this is a simple optimization problem where you have to optimize the degeneracy of states over this thing. You have to optimize this h of k, keeping fixed the resolution and the number of data points. Okay? And what you get is that the points that lie on this maximal curve, they essentially satisfy a power law distribution. And this actually is something that you can figure out with a two-line calculation. Okay, so this is interesting. And because it tells you that, it tells you that the slope, this exponent mu that you have here is related to the slope of this line when you do this two-line calculation. And so you find that there is a special point, which is when mu is equal to 1, because essentially if you are on this side of the curve, when you compress, you get more bits on the generative model than the ones that you squeeze away. If you are on the other side, when you compress by 1 bit, you get less bits on the generative model. Okay? So this point, mu equal to 1, is very special because it's the optimal trade-off between resolution and relevance, where you are sure that you are not losing any bits of information. Okay? And this corresponds to what is called zip flow. Okay? So this suggests that essentially you see zip flow in many systems because essentially you are looking at an efficient representation at the optimal trade-off. Okay? So I think this is nice, but it's not the main subject of today. Okay, so what we did is, to confirm that actually, if you look at now the great thing about deep neural networks and learning machines and machine learning is that actually you can look inside these systems and check whether this is true. Okay, and what we confirmed is that actually, well, also other people confirm that when you look at the distribution of the frequency of states in the optimal representation of restricted Boltzmann machines and deep belief networks, you actually find these power laws. Okay, and the same is true also in efficient representation in minimum description length. So let me just spend a couple of minutes on the issue of now understanding what is the statistical mechanics that corresponds to these internal representations. Okay, and so if you think a little bit about this, then what you find out is that these learning machines are very different from physical systems. In the sense that for physical systems, we know how things go. You have a physical system that is in equilibrium with a certain environment, and you know that the state of this system, so the Hamiltonian is given. You have a certain Hamiltonian which tells you what is the energy that corresponds to a certain configuration. And then the distribution over the energy states is given by maximum entropy. Okay? So essentially what this tells you is that any microscopic state of the environment, which has any typical microscopic state of the environment, which has essentially the same minus log of the probability, is mapped into a state that has more or less the same energy. Okay? So the distribution of energy levels is very narrow. Okay? It's peaked. And this is essentially what the asymptotic equipartition property tells you. And if you work a little bit, what you find out is that the number of energy levels at this level is proportional to the exponential of the energy, okay? Now, if you look at learning machines, actually the situation is very different. Because in a learning machine, actually, what the system cares about is coding, is a representation. So the relevant variable is how many bits you need to code, to use to code a particular state, which is minus the log of the probability. The probability, in some sense, is given by the data, is given by what you want to represent. What you really want to optimize on is on the Hamiltonian. So you want to mold the Hamiltonian, to change the Hamiltonian, the energy levels, in such a way as to achieve an optimal representation. And to cut a very long story short, but I can give you some hint about this. So the principle, if you look at an ideal machine that wants to get an efficient representation, it should be a machine such that the entropy of the energy distribution, of the distribution of energy level, should be maximal. So this tells you that the distribution, the degeneracy of energy levels has to be exponential. Okay, it's again a two-line calculation. And this conclusion here is essentially the same conclusion that I draw for a single sample if you just substitute for P just the frequency. Okay?
But that's the same conclusion as above, that the multiplicity of a given energy has to be exponential.
Exactly. The only thing is that this law here applies on a single energy. Instead, in a learning machine, what you want to have is a broad distribution of energy levels. Okay, so that this law applies on a broad range of energies. Okay, and this is actually what you find in learning machines. Okay, so you want that the system behaves as a physical system on a subset of the examples that you classify in the same way, okay?
But in the learning machine, eventually what you want is to maximize the log likelihood, which is equivalent to minimizing the energy. So then you don't care that in the landscape there are a lot of energies. Eventually you want to get to the minimum in the trained learning machine, not in the untrained learning machine.
Okay, so you are talking about the energy as a function. No, you are talking about the energy as a function of the couplings. You are not talking about the energy as a function of the internal representation. So what I'm saying, yes, so that there is a goal function that is optimized. So this, so if you take a receivables machine, then so you have internal layer S. This is the data visible layer. Here you have all these connections. These are the J's, if you want. Okay. So what I'm looking at is at the minus log probability that you of a particular state of the internal machine after you learn. Okay, and this is a function of the J's.
That
okay.
are fixed because you have learned them already.
Exactly. Okay. So and what I'm saying is that essentially the distribution of this thing, and this is something that you can observe. I mean, during learning, actually the distribution of energy level gets broader and broader.
Over the states.
Over the states, yes.
Okay.
It gets broader and broader. And so because essentially you want precisely to distinguish what is typical of your data set from what is very atypical, okay?
This is the property of the data set, though, right? This is the property of the data set. There's nothing in... If your data
it is, yes, indeed, yes. This is another key point. I mean, you get this broad distribution if the structure in your data set is rich enough. If you're learning noise, then you don't, okay? Or you I mean, you will get a broad distribution, but it's just learning noise. Okay.
What if you have a very low dimensional signal, like you have 1 point? Right?
Sorry?
What if you have a very low dimensional signal in your data? I mean, your data should be rich in the sense.
Yeah.
There is a rich enough representation hidden in the data.
Yeah, exactly. So any, let's say, any data will have some structure, and here the point is, at a certain point when you look at a compressed enough representation, you will see this structure emerging in the internal representation, being, say, mapped into the internal representation. Probably another way to see this is the following. So we think of structured data sets as data sets that contain features, okay? So there are some hidden features such that if I look at my data points just plotted just as just data, then I just see a cloud and I cannot see really differences. But if I knew that there are some features which are some function of the data that are relevant, okay, then I would see a structure, okay, low dimensional manifolds and all these things, okay. So so how does this picture is represented into the internal state of your machine? Well, your machine is just mapping the states into the internal representation, and so you should have a function which is defined on your state that essentially approximate your features, okay? And if these are really all what you need to define the structure of your data, then it must be that these are the sufficient statistics of the model of the internal representation, okay? So it means that conditional on fixing a certain value of the average of these sufficient statistics, then everything else is noise. So everything else is maximum entropy, okay? So it tells you that the probability distribution on the states of the internal machine given a certain average feature should have this exponential form, which essentially in the end, where these G are obtained by solving these equations, which in the end tells you that the probability distribution over states must be a function of the features. So this tells you that the amount of information that your internal representation has on the features is lower bounded by the entropy of the energy distribution, okay? So this is just by data processing inequality. So, which means that if you have a machine with a broad distribution of energy levels, this has a large capacity to store features, okay? Okay? Okay, so this is a little bit the first part, the general part. So how do we apply this thing to neural data? And this actually is what essentially Ryan did. So I'm just, he really did a remarkable job here. So essentially the problem is when you look at recordings from single neurons, then you just have a sequence of zeros and ones. And the question is, some of these neurons, maybe grid cells, may correspond to something which is very much related to behavior, and some of these may not be so much related to the behavior you are interested in. So how can you tell these two things apart? Okay. Of course, I mean, if you have the behavioral correlate, then you can do this plot and you can make this distinction and so on. But in the end, all the information should be in this sequence of zeros and ones. Okay, you should be able to answer that question just by looking at the sequence of zeros and ones. Okay, so and also this is also interesting because you want to answer this question even without knowing what is the behavioral correlate. Okay. Okay.
You're assuming somehow that the set of neurons that you've collected, to the extent that they have an expression on some behavioral correlate, it's shared across the neurons, because otherwise each one could have an entirely different behavioral correlate that you've not observed.
Yeah.
There'd be no way to tell.
Yeah.
Is that right?
Okay. So
There's some
of course,
commonality
the
across
data
neurons
that you study are always the output of carefully designed experiment where there is a behavioral correlate which is supposed to be. But we don't take this assumption, okay? We only say we only look at this time series and try to get with a number that tells you this neuron is doing something relevant or it is not.
The relevance now
Relevant for
will be
what?
defined by a relationship with the other neurons.
No,
No.
No?
it's internally defined just from the sequence of zeros and ones.
So I could do this with one neuron and decide whether it was relevant or not.
Exactly, yes.
Because there are different questions. If you remember this V1, V2 example that I discussed, this communication subspace, asking what is the relevant directions for covariance in V1 is not the same as asking which directions are relevant for triggering activity in V2. So to
Sure,
look
but
at the eigenvectors of the covariance in the neuron population is not going to tell you what is relevant for an external covariate.
That's right, which is why I was suggesting it was an assumption. But the point is that if I just have an arbitrary string of zeros and ones, there may be a behavioral correlate that has exactly that string of zeros and ones, and now it's
Yeah.
perfectly
How
relevant.
do you know if you know nothing about the
So I don't understand where the assumption is that allows you to do
I
this.
guess Matteo is going to
No,
tell us.
well, I'm not sure I understand your question. So if your question is, I can cook up a counterexample where essentially I would get, say, a very relevant thing, but it's actually not doing anything, yes, you can do. Okay, but here we're looking at real data.
The question is relevant with respect to what? And
No.
that's the question.
Yes, that's the issue of featureless inference. It's relevant. So he knows what is. He knows the relevance. I mean, the neuron knows the relevance, and we have to figure
The
out.
counterexample I'm concerned about is actually the opposite one where you think the neuron has no relevance,
Yeah.
but in fact it does, because there happened to be a behavioral sequence that was that had corresponded to exactly the zeros and
Yes.
ones.
So
that you had.
I'll get to this point. This is
Okay.
an open issue in the sense that yes. So here the point. The way in which we are doing this is essentially if you divide this time series in bins of dt, then essentially this you can define a state for each of these windows. Okay, and so the state will be essentially a time stamp at the resolution dt. Okay, so you can look at a particular neuron, how many times it fires in this window, and the frequency will tell you what is the probability that neuron fires exactly in this time window. Okay, now I can compute the resolution and the relevance, as I told you before. Okay,
So I'm not sure what those are here. What is relevance and
so this
what
is
is K and what is S?
okay. So these are the two things I defined before.
But in this example,
In this,
Before you were
it's exactly the same thing as before. So, S is the bin where the spike train occurs, and then you compute how many times that neuron fires in bin S, and then you compute how many bins, in how many bins the neuron spikes k times, and then you compute these two numbers, okay? So let me give you an intuition of what we are doing here. So this is an information on the temporal resolution. It tells you at what resolution you are looking at the data, okay? This is telling you at that resolution what is the dynamical variability of that neuron, okay? How many different frequencies do you see, okay? Do you see just one frequency? Something, so a neuron that behaves more or less at random will have a distribution which is Poisson, so more or less you see one frequency, okay? A neuron which is instead doing something interesting would have a broad distribution of frequencies, okay? So this is the idea. Why? Because essentially when you do an experiment, you are probing the subject with a particular task, and this is supposed to, say, explore the dynamical range of the internal representation in a, say, reasonable way, okay? So that's more or less the idea, okay?
So dt is the width of that cyan region in your other plot. If you had 10 time bins
Yeah,
of zeros
yeah.
and ones was what dt was, and you're just assuming a count code. So you're ignoring the pattern of spikes within that window.
Yes, so this is the first thing. So now,
I didn't know if dt was
Can
what
I
one
get-
was zero or
okay.
one was, or dt is.
yeah, so you can do this for 1 dt, and then you can change dt. So if you do it for 1 dt, you get relevance for that particular scale. If you change dt, you get relevance for all scales. This is why we call it multiscale relevance.
So I
Okay.
guess you count spikes in windows of size dt, and you compute the entropy of that count.
Exactly, exactly.
What's one number? Is that hk or that's hs?
You compute the entropy of the probability to that neuron spikes in S, and this is h of S, and you compute the entropy of the frequency distribution. The probability that if you take a spike at random, that will belong to an interval where the neuron spikes k times. It's a distribution of, so this is related to the distribution of firing rate, k, okay, the number of times the neuron fires at that particular resolution, and this is related to time, okay, to the probability of how uneven is spike, how unevenly are the spikes distributed, okay?
Within the window?
Hmm?
Yeah.
Across the time, across the.
I'm confused, I'm sorry.
I think you.
So,
I see one entropy, but I don't see what the other one is. I just don't.
One is
yes,
the entropy of the spiker, and the other is the entropy of the counts of the spiker. in general.
Yeah.
How variable.
How
Yeah.
does the entropy of the count of the spike
Well, you
itself?
just make a histogram. Of
so you have your,
Spike words.
so this is your time interval dt. You call this S, this is S plus 1, etc., S minus 1. Then you count how many times the neuron spikes here. This is Ks, okay? Then an estimate of the probability that that neuron fires in this bin is just Ks divided by n, the number of spikes, okay? Then I also compute an estimate of the probability that if you take a spike at random, this will belong to an interval where the neuron spiked k times, okay? And this is k times the number of intervals where it spikes k times divided by n, okay? Now you do p log p of this, you get h of s. You do p log p of this, you get h of k, okay? Thanks for I think this is important because, okay, you do it for one particular time interval, you get 1 point. you change the t, and you can go from very high resolution, where essentially every spike is in a different interval, to very low resolution, where all the spikes are in the same interval. Okay, and there is a curve, and you compute the area under this curve, and this is what we call multiscale relevance.
Are you going to tell us the
Multiscale relevance.
Yeah, that's.
Are
Yeah.
you going to tell us the dt where you get the nice mu relationship? Is that where this is going?
That's a very interesting question. I don't have the results on that. We have looked at this, but it's not in the paper. Yeah, yeah. So I wish Ryan could be here to tell you more about this, but this is an interesting thing. So yeah, so you see here are two neurons. One is a grid cell, one is an interneuron, and we say this is more relevant than this. And indeed, well, this corresponds to a very nice firing pattern, and this instead does not. So it's not relevant according to this measure. Okay, now if you look at, so for each neuron you can compute this number that we call multiscale relevance. And you can compute also other numbers, like this is the spatial information, mutual information with the position of the rat. This is, you might have understood, this is on the data set from grid cells, okay? And what you see is that all the neurons that have low multiscale relevance do not contain any information on either the space or on the direction of movement, on the head direction. Whereas all the ones that have a high information content, they turn out to be relevant. Okay. So these are the top 5 neurons up here, and these are the bottom 5 neurons up here. So you see that grid cells, many of them they have high relevance, but there are some of them which are not that relevant. And okay, so.
If I can make a comment, I mean those ones that are the grid cell that has low relevance, if you look at the firing map, it's not really a grid cell. Because to identify something being a grid cell, people use this grid score and this kind of things that are very suboptimal. If you look at it, it doesn't look anything like hexagonal, or it's just classified as grid cell because the measure of grid cellness.
Yeah, so this is, Ryan went on to this other very nice data set that was mentioned by Ben, and looked at, computed the multiscale relevance as a function of, say, behavioral correlates for many, many other examples. And this result that low multiscale relevance correspond to neurons that do not contain information on essentially anything relevant, actually stays there. I mean, is confirmed in all the cases that we have looked at. Here there are different measures like head direction, mutual information, selectivity with respect to head direction, and yes, these are the same, but for neurons in ADN and in postsubiculum, and these are also other measure how this multiscale relevance compare with mean vector length and head direction selectivity. So what we find is generally that there is an increasing trend, but the multiscale relevance actually cannot be explained by any other measure. So it's really an independent measure. Okay, this is again comparing. So for example, grid score is not very much correlated with multiscale relevance for probably the reason that Ryan just said. And different measures have a different degree of correlation with multiscale relevance. So,
can I just ask
yeah.
on this? So it seems like what you're calling relevance here is sort of a measure of clumpiness of the spike tree. And so the assumption seems to be that the correlate that it is relevant with respect to is something that itself has a certain sort of spectrum of variation in time. So that, for example, position is going to be, have an autocorrelation with a particular time scale because you're doing this with respect to time. And that's why something like a grid cell would show up as being relevant. But if I were to do this in the visual cortex and have a very rapidly changing visual environment, then I may not get that temporal persistence at all. So
okay, that's
that what you're
very
defending?
interesting because, and that's one of the problems of this measure because we don't take into account, okay, so we take into account this temporal dimension very mildly in the sense that if you think about it, when you compute this multiscale relevance, you treat the data as really IID. So you don't really take into account that the order of the spikes of where this.
But your bins are always temporally contiguous, right?
Yeah. So we take into account
that's the
that
scale,
thing
yeah.
At a particular scale,
Yeah.
and then we look at that
After
overall
that, it's
scales.
IID.
Yes.
At
In
each
this
scale, the question
yeah.
is how clumpy are
Yeah,
is the
yes.
distribution.
Concerning the issue of burstiness, or say, that's true. Essentially, what we say is that relevant neurons are those which have a broad distribution of frequency, and we have compared this with. Maybe let me just go to this. Oh no, well, I'll get to this. We have compared it with different measures, and with all the measures that we have, like I'll show you a result on local variation, which is also doing something like this, or burstiness, etc., etc. In all cases, we find that MSR explains more than any of these other measures do. Okay, so, and I think it is because all these measures in the end are parametric, or they are making some assumption on on what is relevant, say pairwise correlation, or say the statistics of interspike statistics, etc., etc.
Okay.
Yeah. So let me. So this again. In the same thing. So what we find is that this multiscale relevance decreases with the firing with the activity, neural activity. This is log number of spikes. But also, if you look at the residues along across this thing, you still see that there is a lot of significance in the sense that those which have a high residual MSR are those that are contain information on either space or head direction. And if you take now, you can identify the top 20 neurons, most relevant neurons, and you can identify this only by looking at the spike train. Okay. And then you ask yourself how well you can decode the position of the animal. And you do this. You can do the same exercise by looking at the 20 neurons which has the highest mutual information with space, or for which neural activity has highest mutual information with space. And you see that essentially the top relevant neurons do as well in decoding both space and head direction as if you take the top informative neurons. This is interesting because essentially in order to find these top 20 relevant neurons, you don't need to know the space. And that's exactly the point. I mean, that is supposed to be a region where these neurons know the space, know the position, okay? This is about coefficient of local variation. This was one of the referees asked us to check for this. And you can see that indeed the MSR is positively correlated with this coefficient of local variation. But if you look at the residual on this curve, you see that still there is some information in the MSR that is on the space or on the head direction, which is not contained in the local variation, coefficient of local variation. So, and on top of that, the top 20 relevant neurons, they code position much better than the top 20 neurons according to local variation. So it's not only local variation, it's not only, but it's really well, something more, yeah.
What about firing rate, if you make the same plot with most active neurons?
That we didn't do. The most active ones. Generally, I would imagine you get a very bad result because you would get interneurons, which are very, very active, but they are integrating the signal of other more specific neurons. And so
you showed it's correlated with activity, right?
Yeah. But so the very active neuron, you see these are interneurons. And their firing map has very low information on space or head direction. Okay, because you know much more about interneurons than I do. So there are a number of questions and open issues. So I think this is just exploratory thing. So for example, yes, we find a lot of neurons that are relevant, but that are not, that have no they are not coding for space or head direction. So what they are coding for, okay? So we see that essentially so one exercise that we did in the paper is to take a neuron to estimate the rate map, and then if this neuron would just code for space, you would expect that if you resample the rate map from, if you take a spike train from the resampled rate map, then you should get an MSR which is more or less the same. And this is true for essentially the neurons which contain a lot of information about space. But then there are other neurons that do not contain information about space, that may contain information about something else. So another question is, can we extract more information from these curves? Here we just extract one number, which is the area under this curve. One question is, yes, there is one point here where the slope is 1. This should give you an estimate of what is the time scale at which these neurons are active. Actually, there is more information because it's not only the slope, but it's also the curvature. For example, we find that interneurons have a shape which is remarkably similar one to the other, and the slope is rather constant, as if they were translating, resolution into information on the generative model at a constant rate over a broad range of time scale. So these are questions on which we got some result, but nothing really that we could, yeah. So, and you see, for example, that, so this is an example where you see that grid score, these are two grid cells, this is what Yasser was saying. There are two grid cells, the grid score is the same, but their mutual information in space is very different, and also their multiscale relevance is very different. One is very relevant and the other one is not. Okay, so the other idea is how do you extend this to from single neurons to many neurons. We have explored, say, pairs of neurons, looking at Boolean functions of neurons, the or and the and. It looks like there is not much information in the correlation, say, so for example, if you look at the OR of two neurons, or if you look at the XOR of two neurons, you essentially get the same multiscale relevance. Okay, so looks like these are behaving like independent neurons. Okay, so this is the summary. So as I told you, we believe that in order to understand these complex systems, we need something which is what we call featureless inference. And then essentially information is carried by the frequency distribution and can be quantified by the entropy of the frequency distribution, which we call relevance. And these maximal informative samples in this sense, maximally relevant samples, exhibit statistical criticality, which we think is interesting because it gives you a general explanation of why you see this broad distribution everywhere. And then we use this to characterize the temporal structure of neuronal activities. And it looks like, say, this measure contains all information on relevance that you can get with other measures, but it gives you more information. And also there are a number of interesting questions, including the fact of how you can extend this approach to other systems. Okay, and so we are currently working on this. Thank you very much. Yeah, please.
So you've called it featureless, and I guess I think I understand what you mean by that.
It seems like you need, there's a kind of a non-exchangeability that's crucial in the data here, right? If I took the spike trains and just took 1 millisecond bins and then reordered those bins arbitrarily, I'd lose all the information.
Yes.
So what it seems like what you're leveraging is the idea that you have, your data have arrived with a label, and whatever the feature is, even though you're not committing to the nature of the feature, it varies with some property along that label. In this case, time. And for DNA methylation, it would be location along a chain.
Yes, yes.
And so I guess the question then becomes, what is that property? Can you actually put your finger on the nature of the correlate, of the covariate, or the behavior of the covariate that is necessary to do this?
That's exactly the question. So what I can tell you is about a failed project, because I think time is really special, in the sense that what we tried to do at the beginning was to say, okay, why don't we compute the same thing over space instead of over time? So we just divide our box into many bins and then do essentially. You don't get grid cells of relevant neurons. You don't really get. And I think this tells you that really time, time is what all you have in the brain, in the sense that everything you have to represent it in neural activity.
But it's more, but the covariate has to.
What puzzles me about this comment is that if I scramble this over time, I will get the same P hat of S and the same P hat.
No,
No,
no.
Not if you,
You get the
not if
same
you do it
P-
in the sub bins. If you
No,
do
no,
1
no.
millisecond bins and then your
Yeah.
bins.
No, but
No,
if
no.
I scramble the cells, if I have
Yeah,
decided
once
cells
you
of a given
once
DT
you
at
grab
a
the
given
cells, yes.
resolution,
Yeah, but that's why you
Once
scale
you
over
grab
different
it.
resolutions.
or only one at a time. Say that again.
So the thing is that it's true. If you get a fix the bin size, and then you scramble,
Yeah.
then fine.
Then
But the
if
thing
you
is that
have
we
some
that's
PFK
why we
cells-
measure over different resolutions.
Okay.
Right, but that's
So-
not scrambling.
Huh?
This doesn't
No,
change the
no,
wall,
no,
that's just
no,
getting a
okay,
bigger
so-
window.
Yeah, but then the values, the h of k's change in the sense that so if I fix a time bin,
But what I'm trying to say, Yasser, is that you have a curve. For every resolution that you
Yeah.
choose, the h of s and the h of k, that is to say the point in which you are on this curve, is invariant under scrambling of time once I have fixed
No.
the resolution.
when you have fixed
No,
the
no, no.
time.
Yes, every
If
point
you
is-
scramble time,
No, if I scramble
inside
bins,
here-
if I scramble bins, I fix DT.
Order of the bin.
yes, you scramble
So
bins, yes,
PFS
yes,
doesn't-
yes.
So in some
Yes.
sense, at that resolution, time doesn't matter.
Yep.
You,
Yeah.
you have the
That-
same point
yes.
in the
That
curve.
resolution, yes.
And now
Yeah.
you know you trace the curve, but every point in that
Exactly,
curve is
yes.
invariant
Yep.
towards
Yep.
a
Yes.
permutation of the bins. So
But this-
time.
No, but
No,
but
but
time is
it-
fundamentally important because it defined the bins.
it
Yeah.
defines a resolution. It
No,
defines
well, it defines the
how
bins.
you
No,
move
but-
around
but-
the
but-
x-axis in this
But you're
curve.
shuffling at
Okay.
at
It's a
a
subtle
given
point.
resolution is a different process than shuffling at a different resolution.
Yeah.
So time is still.
Yeah. So
So
you're right,
cannot
yes,
do
so,
both. You cannot vary DT and scramble at the same time.
No.
That's
Yes.
what you're telling me.
Exactly.
That if I want to keep DT as a variable, I cannot think of
And
scrambling.
and if you choose a very time bin and scramble and then calculate the whole thing, then you
Yeah.
just get nothing.
and also but scrambling bins, it's a very complicated, because you have to put all the time points. But yes, next time we should have you as a coder, so
So-
you can explain.
The other thing that you're saying is that high relevance, which is what you're after, which is a high h of k,
Yeah.
has to do then with a lot of variability, with an almost flat histogram over k.
No, it's not flat, it's power law.
Well, but
It's
if I
power-
wanted to maximize h of k, that would be the way to
No,
maximize.
because you have constraints.
You have the constraint, I
Yeah.
understand.
So with those constraints, the
the
largest h of k that you can have,
This
Is
is
a
what
power
gives you
law.
the Sweeps law.
yeah. Yeah.
So Ilya Nemenman published a paper last year where they showed that groups of 3 spike bursts, if you jitter the time of the middle spike, it carries a lot of information.
Uh-huh.
That you could decode, that basically the thing you read out depends a lot on whether that middle spike is shifted earlier or later.
Uh-huh.
This method would presumably be blind to that. Do you agree? That, in other words, you're only thinking about counts in a particular window.
It depends on how you define your delta t. If your delta t includes all 3 spikes, then the order of the 3 spikes in that delta t doesn't matter. But if I now decrease my delta t, then
Yeah.
which spike falls off and comes into the next interval will depend on how separated they were temporarily.
You'll get 2 and
So-
1 versus 1 and 2.
Exactly.
So-
But the order doesn't matter, as we said, we can scramble the
No,
bins.
the order, no, but as you decrease your resolution, as you increase your resolution, decrease your delta t, then these things will split differently.
So, but
So
what
at
is,
the
is there a
resolution, it won't matter.
particular time scale
That's right.
that happens at a particular time scale?
That's
or-
right. They're looking at like 1 millisecond jitter of the middle spike.
Ah,
I mean,
okay.
granted, this is the motor system, the Songbird
Uh-huh.
motor system, so
Uh-huh.
it's very closely linked to the,
Yes, but-
effectors.
This is refers to multi-spike, multi-neuron recording, or just a
I don't know
single
if it's
neuron?
multi-neuron, this is a single neuron analysis
Is
though.
the
Single
Ilya
neuron
Nemenman
analysis.
Yeah.
paper?
Yeah, this is a,
Uh-huh.
PNAS? I forget where it came out. Anyway, I mean, such timing codes seem rare. I'm a rate coding guy, but it just
Uh-huh.
it seems like this is a measure that's fundamentally committed to like spike count in
Yeah.
bins as being the relevant. So patterns, we think of the old like information theoretic approaches where you count the number. Initially, that's where I thought you were headed, was you were going to count the number of binary words within a DT. If you bin 1 millisecond bins, you have 2 to the
Yeah.
n possible words within there, but you're not looking at any of that kind of pattern information.
No, we are not.
And mostly that probably is fine.
I mean, say, here we are not.
Yeah.
Um, to address this question, I would try to see whether one can use the same principle in a different way.
Instead of just taking account of a bin, you could take
Yeah,
some other statistic.
here, say, we just take the count. We don't take the sequence. But yeah, maybe one could do, yeah.
But I think it's the same thing I was mistaken before. Once you start varying the resolution, then that problem goes away. At the fixed resolution, you have that problem, that you cannot distinguish between 1, 2, 3 equally spaced, and 1 and 2 very, very close to each other, and the third one coming later. You cannot distinguish that.
I don't think this
But now, when
yeah.
you begin to decrease the resolution, you will see the difference between these patterns.
No, because you either get 2 spikes in the left beans or 2
Yeah.
spikes in the right beans, but the order of beans doesn't matter. So
But
you
if
have
you
2
then
in
go
1
to
or
a
1
smaller
in
time
But
bin,
then
that
you
there's
make it
only
smaller so
one,
that you either have 1 or 0 spikes.
But every
But
then you can
But then you can scramble again and it's irrelevant.
You can scramble them.
Yeah.
Yeah. I forgot what I said before. I'm forgetting myself. Okay, good.
Yeah, I was wondering, like, if you can give an intuition of what this relevance measure means. Like, for me now, I think of it, because you're doing this scan of all the timescales. Is it like it's the most relevant across all timescales? Because maybe you can also think that some neurons are very relevant for a specific timescale, but not for other. And does it
Exactly, yes.
then average?
Mhm.
And then do you lose these specific neurons, or how does it integrate?
Yes. So that's a very interesting question. So the highly relevant neurons that we find, they close to saturate what is the maximal value of h of k that you can get, that you can compute, say, theoretically. So there are small deviations. So for example, in this example here, you see that there are neurons, in low relevant neurons, you can see these differences. So you can see that, say, this neuron 10 is more active at high frequency, and this one is more active or is more relevant at low frequency. Okay, so you can do these things, and this is part of looking into the shape beyond this multiscale relevance. Yes.
What is the timescale that gives you the maximum? What you said is the
It varies. It varies. Actually, we have looked at the maximum that corresponds to slope 1 here, that corresponds to this zip flow. And that, well, I have a result I have to remember, but essentially it was giving sort of consistent results in the sense that compared to, say, time scales of grid cells or of other neurons, we find consistent results. But that, yes, that's it. I mean, this is it varies from neuron to neuron. So you can.
Shouldn't it depend on the speed of the animal when if it's carrying information?
Yes, that's one thing. I mean, I can look at these results on these timescales, and we can discuss more. But yes, there is this information.
Last question. Have you compared to integrated autocovariance, which would be a very similar measure but replacing your entropies with variances in effect? Would that give you something similar?
Look, that is a featured this strong opposition towards, because it's based on pairwise interaction, pairwise statistics. Okay, covariance or variance is based on just saying that everything is contained in 2-point functions. Why should it be so?
Well, I'm not saying that it would be, but I think it's an empirical question as to how much it is.
Yeah.
But still pair.
Autocorrelation. You take a point at time t and a point at time.
Yeah, 2 points in time.
So yeah.
But I mean,
So
I guess
for
the
example,
empirical
3
question
point.
is to what extent is it?
Yeah,
right?
what do you gain by going entropy and
Yeah.
Well, just
versus
how
stopping
much
at 2 points
how much
of decrease?
additional
Yeah, how much do you gain?
structure is there in the entropy?
Yeah, yeah. Well, to some extent, local variation coefficient is a 2-point. function is a 2-point statistic, and it takes into account a little bit the variance of the interspike distribution, essentially. So, and yeah, so.
Maybe we can continue discussing on the computer. Thank you very much.
Okay.